This is week 11, newborn screening, treatment of genetic disease in newborns and adults. The learning objectives for this week's work is to discuss general strategies for treating genetic diseases in newborns and adults, to discuss the treatment options available for diseases typically detected by newborn screening, to discuss the successes and challenges, to discuss the rationale for newborn screening for conditions that are not reliably treatable, and to discuss genetic counseling issues related to newborn screening. Let's start off by looking at the state-based newborn screening programs. These are one of the most established population-based public health screening programs in the United States. It serves as a model for genetic screening programs. It's very successful public health initiative, and it has the ability to detect congenital defects before disability and or death. The U.S. government can only make recommendations. It cannot mandate newborn screening. It remains under the jurisdiction of state governments. The states differ in their requirements, and it began with PKU, and today many states screen for about 60 conditions. The Advisory Committee on Heritable Disorders in Newborns and Children, the ACHDNC, issues a recommended universal screening panel, which is referred to as the RUSP, that identifies a number of core conditions, those for which screening is highly recommended, and secondary conditions for which screening is optional. As of November 2016, the RUSP included 34 core conditions and 26 secondary conditions. Please go to this link here to this website and you will be able to view the disorders. The National Newborn Screening and Global Research Center, NNSGRC, serves as an independent U.S. National Resource Center for newborn screening and provides newborn screening information globally. Please visit the link so that you can see what is offered in your specific state for newborn screening. Please remember that it is fluid because new additions will be added from time to time. So let's look how the newborn screening procedure and management should work. A blood sample is taken in the first few days by impregnating a special filter with the blood. It's often taken from the newborn's heel. Now, in the very beginning, when PKU was first tested, it was tested with a bacterial inhibition assay, but today we use mass spectrometry to detect abnormal levels of metabolites. And what's great about this test is it can test a larger number of metabolic disorders. After the test is taken, the sample is taken, it's sent to the state laboratory for analysis. If the lab identifies an infant with a positive newborn screen, the infant's primary care provider is contacted. This individual is then responsible for contacting the infant's family and to provide initial counseling about the disorder, also to arrange a subspecialty referral and to obtain a repeat blood sample, which should be done between 7 and 14 days. Many conditions are extremely rare, and you cannot expect all primary practitioners to know all the details about these different diseases, so most of them are sent to an expert for education and for additional confirmatory and severity testing. However, notification to the parents is not always done as it is supposed to be done, and Allison Wood had a very bad experience with her notification on a weekend when it was a holiday weekend and didn't know about her disease. She had a son who has glutaric acidemia type 1. She read about the disease on the internet, and so did her husband. So I have a link here for you to look at, and this is something about Allison Wood and her family. I met her at the NERG conference in December. She is planning on applying to genetic counseling programs. She became very interested in genetics and is now working with the state of Vermont and putting together some guidelines for how we should identify these conditions and explain them to the parents and not leave them hanging without any information. She also works with the Rare Disease Organization. please visit the NORD, the National Organization of Rare Diseases website, and you can learn more about this great organization. And please note that February 28th, 2018 is Rare Disease Day. It happens every February, and see if that we could do something to get involved with this. Now, the other thing that they would like to do is to set up a student chapter here at BayPath, and it doesn't just have to be the genetic counselors, although it would be nice for us to start this up so that we could have a chapter here. And NORD is the official sponsor of the Rare Disease Day, and millions of patients and families around the world are going to share their stories to promote awareness of the challenges, the hopes, and the needs of those individuals who are living with rare diseases. So please contact me with this if you have some ideas or you wish to work to participate in this. This slide is a picture of two nurses at Bay State Medical Center giving the hearing test to a newborn. Once a child has had a genetic screening test that shows that they are at increased risk for a specific condition, then this is followed up with a diagnostic test or tests that look for evidence of the disease. Now, early accurate diagnosis of a genetic disease can lead to treatment, or it can also lead to enrollment in a clinical trial, so you can test a new therapy. And it provides information to the parents on their risk or risks to future pregnancies. Genetics actually adds precision to a diagnosis. However, we also see that there's a downside of newborn screening in that follow-up tests may not show the abnormality. In such a case, the family may then experience what has been referred to as patience in waiting, when the genotype associated with an illness is present but the phenotype isn't, or isn't yet. The related condition may actually never manifest, perhaps because of effects of other genes. And studies have found that this incomplete knowledge may interfere with parent-child bonding and cause anxiety for years. Please visit this link to an article to talk about one of these problems that are associated with newborn screening and the concept of patience in waiting. What if no medical intervention is possible? Now, prior to newborn screening, most inborn errors in metabolism were diagnosed after the nonspecific symptoms. We discussed some of these last week: seizures, odors, dysmorphic features, failure to thrive. And the presenting symptoms for many diseases are similar. But it's important to remember that the disease progression is not. Some are very fast, and some are not. The challenge with newborn screening is to diagnose and treat as soon as possible before the signs and symptoms manifest. Some states actually allow parents the opportunity to give permission to test supplementary testing for diseases for which there is not an available treatment. Some decision makers believe that the benefits of identifying a disorder without a known treatment outweighs the potential risk or undesired consequences. As a genetic counselor, you need to think about that, because we don't have treatments for some of the diseases that we are testing for in newborn screening. So therefore, with treatment, the tests for genetic diseases currently greatly outnumber the treatments. And the treatments need to correct the abnormality in enough appropriate cells and tissues to prevent or minimize symptoms while not harming other parts of the body. If we look at the treatments for single gene disorders, some of these include removing an affected body part, such as the spleen, part of the liver; diet, changing the diet, stopping people from eating certain things; delivering a transplant from a donor of a body part or possibly a biochemical; delivering human proteins from recombinant DNA technology; refolding correctly a misfolded protein; blocking gene expression; using gene therapy to add normal alleles without removing the mutant alleles; and using genetic and genome editing to replace, delete, or add alleles. Gene therapy delivers working copies of genes to specific cell types or body parts, typically as part of modified viruses that function as carriers, called vectors. More than 2,000 clinical trials of gene therapies have been conducted worldwide since 1990. The first approval of a gene therapy in Europe was in 2012 for a rare enzyme deficiency that causes pancreatitis, but it was discontinued due to the high cost and low demand. Adding functional genes to treat an inherited disease may provide a longer lasting effect than treating symptoms or supplying a protein. A gene therapy may require a single or a few treatments, whereas a traditional drug or even enzyme replacement, substrate reduction, or pharmacological chaperone therapy may have to be administered frequently for the patient's entire life. If we look at two types of gene therapy, we can look at germline gene therapy, which alters the DNA of a gamete or fertilized ovum so that all cells of the individual have the change and the correction is heritable. If we look at somatic gene therapy, it corrects only the cells that an illness affects. It is non-heritable. A recipient does not pass the genetic correction to offspring unless the cells that give rise to gametes are inadvertently altered. Clearing lungs congested from cystic fibrosis with a nasal spray containing a functional CFTR gene is an example of somatic gene therapy. Gene therapy strategies vary in invasiveness. Cells can be altered outside the body and then infused into the bloodstream through a vein. This is called ex vivo gene therapy, outside the body. In the more invasive in vivo gene therapy, the gene and its vector are introduced directly into the body, such as through a catheter inserted and snaked to the appropriate organ. There, the vector must enter the targeted cells. The human DNA be transcribed into messenger RNA, the messenger RNA translated into protein, and then the protein must function enough to halt regression of the disease to improve symptoms. I want to talk about Canavan's disease. It's also called Canavan von Bogaert-Bertrand disease. It's an autosomal recessive degenerative disorder that causes progressive damage to nerve cells in the brain and is one of the most common degenerative cerebral diseases of infancy. And below you can see the brain of a normal patient as opposed to a patient that has Canavan's disease, and you can see where the degeneration has occurred in the interior of the brain. In this slide, which demonstrates gene therapy invasiveness, the cells can be altered outside the body and then infused into the bloodstream through a vein. The technique is ex vivo gene therapy. In the more invasive in vivo gene therapy, the gene and its vector are introduced directly into the body, such as through a catheter inserted and snaked into the appropriate organ. There, the vector must enter the targeted cells, the human DNA be transcribed into messenger RNA, the messenger RNA translated into protein, and then the protein must function enough to halt progression of the disease or improve symptoms. Researchers obtain therapeutic genes using the polymerase chain reaction, recombinant DNA technologies, and other techniques that cut DNA. In the future, researchers and someday clinicians may deliver synthetic genes. The origin should not make a difference. DNA is DNA, and the genetic code is universal. Here are some sites of gene therapy. The endothelium. And in the endothelium, remember this tile-like endothelium that forms capillaries can be genetically altered to secrete proteins into the circulation. If we look at the muscle, the immature muscle cells with the myoblasts, they can give healthy dystrophin genes to may treat muscular dystrophy. The liver, to treat certain inborn errors of metabolism, only 5% of the liver's 10 trillion cells need to be genetically altered. And in the lungs, gene therapy can reach damaged lungs through an aerosol spray. Enough cells would have to be reached to treat hereditary emphysema caused by alpha-1 antitrypsin deficiency or cystic fibrosis. This slide is showing you why those four areas are so good for being targets for somatic gene therapy. The endothelium can secrete needed proteins directly into the bloodstream. Muscles accessible. Remember, it comprises about a half your body mass and has a good blood supply. The liver has many functions, works in metabolism, and can regenerate. And the lungs are easily assessed with a aerosol spray. If you look at some examples of gene therapy, we see that gene therapy has had some problems and successes. As you will see in the following three examples, ADA deficiency, OTC deficiency, and RPE65 deficiency. If we look at a severe combined immune deficiency, SCID, which is caused by an ADA or adenosine diamidase deficiency, we can see where we first did some gene therapy. Toxins destroy the T cells, thereby causing susceptibility to infections and cancer. And the replacement of ADA in individuals genetically deficient was attempted. If we look at our first gene therapy patient, of which we had some success, we can look at Ashante Ashi da Silva. In the late 1980s, Ashante's parents did not think that a little girl would survive her near continual coughs and colds. She was always sick. She could walk only a few steps before becoming winded. Ashi had severe combined immune deficiency due to ADA deficiency. At age 2, she began enzyme replacement therapy, but within 2 years it stopped working. She would likely die of infection if nothing else was done. Then her physician heard about a clinical trial of gene therapy that would give her white blood cells functional ADA genes. On September 14, 1990, at the NIH, Ashi received an intravenous infusion of her own corrected white blood cells. She did well. Today, a longer-lasting form of gene therapy alters stem cells instead of the more specialized T cells. It was approved in Europe in 2016. In this slide, you can see the pathway for the severe combined immune deficiency due to the ADA deficiency here. Without the ADA, you build up the deoxy ATP. It destroys T cells. The B cells are not activated. And then without your B cells working, if you remember back to the immune system, you would not have humoral mediated immunity or antibody mediated immunity, antigen antibody mediated immunity. And if we look over here, we see that when you do have the ADA, what happens is you can break it down and it eventually becomes uric acid. If we look at OTC, ornithine transcarboxylase deficiency, we see that the deficiency of OTC is inherited as an X-linked recessive mutation. OTC normally breaks down amino acids which are present in protein. If you have a lack of OTC, it allows the buildup of ammonia, which damages brain function. And low protein diets and ammonia binding drugs are used to treat OTC deficiency. I would like to look at a gene therapy trial which was not successful. Jesse was diagnosed at age 2. He had a mild case because he was a mosaic. Some of his cells could produce the enzyme. At age 10, Jesse's diet lapsed and he became very ill. Years later, after he went into a coma in December in 1998 from missing a few days of medications and recovered, he volunteered for a gene therapy trial his doctor had mentioned. On September 13, 1999, a trillion adenoviruses carrying the functional human OTC genes through a tube into Jesse's liver were infused. But the viruses entered not only the hepatocytes as intended, but also the macrophages that alert the immune system. Jesse's organ systems began to fail. 4 days later, he was brain dead, and he was taken off of life support. This is actually a picture of Jesse, as well as you can see the metabolic pathway by which the ammonia needs to be broken down, and how if it builds up and the ammonia builds up, it can cause mental deterioration. I would like to look at the third example of gene therapy. It was used for Leber's congenital amaurosis type 2. It is a very severe disease. It's the most severe form of blindness. It's inherited as an autosomal recessive disease. It's a mutation in a gene called RPE65. Gene therapy was first tried on the breed of sheepdog that has the same mutation as humans. It was then tried on 4 young adults and later on an 8-year-old, Corey Haas. And today, several hundred people who had lived in the dark shadows or total blindness can now see after the experimental gene therapy was done. What happens is at the backs of the eyes, in the retina, an individual who has this disorder cannot make an enzyme necessary to convert vitamin A to a form that the rods and cones can transmit light energy to the brain and use. And the gene therapy supplies the gene that encodes the enzyme. This slide is showing you what the retina pigment epithelium, the rods and the cones of the photoreceptors that are found in the eye. This table has been included to show you some gene therapy concerns, some scientific as well as bioethical concerns with gene therapy. The genome editing tool CRISPR-Cas9 may one day replace gene therapy because it is much more precise. It not only delivers a gene to a specific part of the genome, but can replace a gene, remove a gene, or add a gene in multiple places, or insert, correct, or delete multiple genes. Its applications in diagnosis and treatment are just beginning. Please go to this video and look at how CRISPR works. At our March meeting, we will be having a presentation on CRISPR and all the newest implications that it has for genetics. This week we will have quiz 3. Quiz 3 covers the material that's in weeks 8 through 11, and we will not be having a discussion this week.